All right, let's dive into the incredible story of gene editing. We're going to trace its evolution from a tool that was kind of like a molecular hammer to one that now works with the precision of a fine-tipped pen. And believe me, that shift is changing everything we thought was possible in medicine.
I mean, this was the dream, right? This was the revolutionary promise that shot CRISPR into the global spotlight. The idea that we could finally open the book of life, our own DNA, and just correct the typos, fix the tiny errors that cause these devastating diseases. It was a level of control that honestly felt like pure science fiction just a couple of decades ago.
But here's the twist. While the dream was all about elegance and precision, the first tool we actually built to do it, well, it was more of a blunt instrument. Incredibly powerful? Yes. A total game changer? Absolutely. But it came with a kind of brute force that could sometimes do as much harm as good.
So let's start right there with that original tool, the one that kicked this whole revolution off, the CRISPR-Cas9 system. This is the OG, the first gen tech that proved that editing our genes on this kind of scale was even possible. You know, the way it works is actually beautiful in its simplicity. It really just has two main parts. First, you've got this thing called a guide RNA, which is like a tiny genetic GPS. Its whole job is to home in on the exact 20-letter sequence in our DNA that we want to edit. And once it locks on, the second part, an enzyme called Cas9, comes in and acts like a pair of molecular scissors, making a clean cut right through the DNA. Now, that cut is absolutely critical. It's called a double-strand break, or a DSB. And you have to understand, to a cell, this isn't some minor snip. This is a code red, a 5-alarm fire. It's a catastrophic event that basically screams that the entire genome is at risk. So the cell panics and immediately slams the big red button for emergency repairs. And this, right here, is the central paradox of that first-generation CRISPR. All of its therapeutic power comes from making that cut, but so do its biggest dangers. Because when the cell panics and tries to patch things up in a rush, things can go very, very wrong. So you might be thinking, okay, a simple cut seems pretty effective if you just want to turn off a bad gene. Why is that cellular panic so risky? What actually happens when that emergency repair crew makes a mistake? Well, the fallout can be pretty severe. In its mad dash to fix that break, the cell might accidentally just delete huge chunks of DNA. Or, even worse, it might stitch the wrong ends together, causing parts of completely different chromosomes to swap places. That's called a translocation. This kind of damage can trigger the cell's self-destruct sequence, or scarier still, it can end up selecting for cells that have faulty safety systems, which is one of the classic hallmarks of cancer. So to solve this huge problem, scientists knew they needed a way to make edits without making that dangerous cut. And that led to the second generation of gene editors, a truly breakthrough technology called base editing. Think of it less like scissors and more like a chemical pen. Instead of cutting the DNA, base editors use a modified Cas9 that just finds the right spot. But then a totally different enzyme comes in and chemically converts one DNA letter or base into another. So for example, a cytosine base editor can turn a C into a T. An adenine base editor can turn an A into a G. It's literally like using a pencil to carefully erase a single letter and write the correct one right in its place, all without ever tearing the page. But of course, that incredible precision comes with a trade-off. Base editing is amazing for fixing diseases caused by one single wrong letter, but it's limited to just those specific swaps. It can't add new letters or delete them, which you need to be able to do to fix a whole lot of other genetic diseases.
And that limitation brings us to the next evolution, prime editing. Now, this is the technology that gets us unbelievably close to that original dream, a true genetic search and replace function, just like in a word processor.
Honestly, the way this works is just so elegant. First, a special guide RNA finds the target. Then, the Cas9 enzyme doesn't make a full cut. It just nicks one of the two DNA strands, so no dangerous double-strand break. Then, another enzyme uses the guide RNA itself as a template to literally write the corrected sequence directly into the DNA at that nick. And finally, the cell's own natural repair systems come in and finish the job, seamlessly integrating the brand new edit.
And the precision here is, well, it's staggering. Early versions had some issues with efficiency, but the latest improvements have brought the error rate down to ridiculously low levels. We're talking as few as one single error for every 543 edits. I mean, that is the kind of accuracy that starts to make gene editing a real possibility for the clinic. Okay, so let's just take a step back for a second. We've seen the three major tools, the scissors, the pen, and now the word processor. Let's put them side by side to really appreciate just how far and how fast this technology has come. This table just lays out the whole journey perfectly, doesn't it? We started with CRISPR-Cas9, super powerful but pretty risky. Then we got base editing, much safer because it avoids the cut, but also less versatile. And now we have prime editing, which gives us both that versatility and incredible precision. It's just a massive leap forward in both what it can do and how safely it can do it.
And to put that safety improvement in perspective, just look at this. Remember one of the scariest side effects of the original Cas9? Those accidental deletions of huge chunks of DNA? Well, with the newer base and prime editors, that kind of catastrophic error is about 20 times less likely to happen. That is a huge reduction in risk. But the quest isn't over, not by a long shot. There's still this shadow that hangs over all these technologies, off-target effects. This is when the editor makes a change at the wrong location in the genome, simply because that spot looks kind of similar to the real target. Minimizing these mistakes is really the final frontier in making these therapies perfectly safe.
But before we can really appreciate just how amazing this cure is, we have to understand the diseases it's targeting. We need to talk about the heavy, lifelong burden of sickle cell disease and beta thalassemia. Yep, this quote really gets to the heart of it. This isn't just some clinical description. This is the day-to-day reality for millions. A sickle cell crisis is this brutal, unpredictable cycle of pain that can just completely take over a person's life.
So you've got these two different diseases, but they share a common, devastating problem. Both are caused by a faulty gene that messes up your adult hemoglobin. With sickle cell, your red blood cells get warped into this hard sickle shape, and they create these agonizing traffic jams in your blood vessels. For beta thalassemia, your body just can't make enough healthy hemoglobin, which means patients are locked into a lifetime of getting blood transfusions every single month just to stay alive.
So the problem seems incredibly tough, right? But the solution scientists found is, well, it's just so elegant. They didn't have to invent something totally new. They figured out how to reactivate a powerful tool that's already hiding inside every single one of us.
It turns out we all have a gene for this perfect high-performance version of hemoglobin. It's called fetal hemoglobin, and as you can probably guess from the name, it's the stuff we all use when we're in the womb. It works perfectly. The only catch is that our body has a switch that turns that gene off right after we're born.
And this is the key, the big aha moment. Scientists found the exact gene that acts as that off switch. It's called BCL11A. And this led to a truly revolutionary idea. What if you could just break the off switch? Could you trick your body into making that perfect fetal hemoglobin again for the rest of your life?
And that is precisely what Casgevy does. It's a three-step dance. First, you harvest a patient's own blood stem cells. Second, and this is the magic part, you take those cells to a lab and you use CRISPR to go in and snip the BCL11A gene, breaking that off switch. This all happens ex vivo, which just means outside the body. Finally, you infuse those newly edited, superpowered cells back into the patient, where they can set up shop and build a brand new healthy blood system.
Okay, so the theory is beautiful, but did it actually work for patients? Well, the results from the clinical trials, they weren't just good, they were absolutely transformative.
Just look at this number, 97%. Out of 30 patients with severe sickle cell in the main trial, 29 of them were completely free of those horrible pain crises for at least a year, many for much longer. This doesn't just manage the disease. For these people, it effectively ends it.
And this chart just says it all, doesn't it? On the left, you see patients were averaging about 4 of these severe, land you in the hospital pain events every year. And on the right, after treatment, 0. A flat line. The pain is just gone. It's a life given back.
And the story for beta thalassemia was just as incredible. 91% of patients were able to completely stop their monthly blood transfusions. Can you imagine the freedom? No more spending days in the hospital every month, no more complications from iron overload. It's a total game changer.
So the gene edit itself is precise, it's elegant, it's honestly beautiful. But, and this is a huge but, the journey a patient has to go through to get that cure is anything but. This is where the miracle meets a really harsh reality.
So, what's the catch? Because there's always a catch, right? Well, unfortunately, yes. The CRISPR part is like a molecular scalpel, incredibly precise. But to prepare the body for that surgery, doctors first have to bring out a sledgehammer.
And here it is. For those new, edited stem cells to have a place to grow, you first have to get rid of all the old, faulty ones. The only way to do that is to completely obliterate the patient's existing bone marrow using high-dose chemotherapy. It's this blunt, toxic procedure you have to endure to make way for the elegant cure.
And that chemo is punishing. The trial data shows it. We're talking about extremely painful mouth sores that make it hard to even drink water, dangerous fevers at a time when your immune system is completely wiped out, and platelet counts that drop so low it puts you at serious risk. It is a grueling, months-long ordeal.
And maybe the most difficult part of all, this chemo has a very high risk of causing permanent infertility. So you're asking patients, many of them young adults or even teenagers, to make an absolutely gut-wrenching choice. Cure my disease, but potentially lose the ability to ever have biological children. That is a profound, heavy price to pay.
But here's the hopeful part. Casgevy is just the beginning. It's chapter 1. Its success has basically kicked off a global race to build on this amazing breakthrough, to find ways to keep the miracle but drastically reduce the price.
You can already see the next wave of innovation happening. So we have Casgevy, the trailblazer. Then you have therapies like EDIT 301, which uses a slightly different version of CRISPR that might be even better at flipping that fetal hemoglobin switch on. And then you get to Beam 101, which uses a newer technology called base editing. Now this is really cool. Instead of cutting the DNA double helix, it works more like a pencil and eraser, chemically changing just one DNA letter. The hope is that this could be a lot safer.
You know, when we think about the gene editing tool CRISPR, we usually think about fixing things, right? Correcting a broken gene that's causing a disease. But what if its most powerful use wasn't just fixing, but upgrading? We're diving into how CRISPR is being used to literally supercharge our own immune systems, turning them into an elite engineered army to fight cancer. So instead of thinking of CRISPR as a mechanic fixing a faulty part, in the world of oncology, it's way more like a military engineer. This is all about upgrading our internal defenses, making our immune cells smarter, stronger, and way more lethal against cancer than nature ever made them.
Okay, so to really get where this is all going, we first have to understand what's happening right now. And the starting point for this whole revolution is a treatment called CAR T-cell therapy. It's a massive breakthrough that's already a game changer for blood cancers. You can think of it as a living drug.
So how does this whole thing work? Well, the concept is actually pretty brilliant. Doctors take T cells, the soldiers of your immune system, right out of your body. Then, in a lab, they engineer them. They add a special new receptor, that's the CAR part, and it acts like a heat-seeking missile, guiding these T cells directly to cancer cells. Then, they infuse these new super soldiers back into you to hunt and destroy.
Now, this is the really crucial part. The way we do it today, the standard approach, is what's called autologous therapy. That means the T cells come from the patient themselves. It's super personalized, which sounds great, but it has some major drawbacks. The future, the thing everyone's working towards, is allogeneic, using cells from healthy donors to create a ready-to-go, off-the-shelf treatment. So what's wrong with using the patient's own cells? Well, there are a few huge hurdles. First, it takes weeks to manufacture these personalized cells. That's time a really sick patient might not have. It is also wildly expensive. And maybe the biggest problem of all, if a patient's T cells are already worn out from chemotherapy, which they often are, the whole process can just fail.
And this is where CRISPR finally steps onto the stage. That dream of an off-the-shelf therapy, something that's ready to go on demand, made from healthy donor cells, well, that's only possible because of CRISPR's incredible precision. It is the key that unlocks this entire revolution.
But wait a second, this raises a massive red flag, right? If you put someone else's immune cells into a patient, wouldn't the patient's body just reject them? Or even worse, couldn't the donor cells attack the patient's body? That is the fundamental problem that absolutely had to be solved.
And this is how CRISPR solves it, with a really elegant, multi-step engineering process. First up, scientists do what's called a TCR knockout. They use CRISPR to basically delete the donor cell's original programming. This is absolutely critical, because it stops the new cells from attacking the patient. Second, they do an MHC class I knockout. You can think of this as giving the donor cells an invisibility cloak. It hides them from the patient's immune system so they don't get rejected. And finally, they can add potency enhancements, knocking out genes that act like safety brakes on the T cell. This keeps these engineered soldiers fighting without getting exhausted.
And look, this isn't just a cool idea on a whiteboard. The most important part is that it actually works.
We're seeing data from clinical trials showing that these heavily engineered, off-the-shelf cells are just as powerful as the personalized ones. And get this, in some cases, they've worked for patients who had already failed other advanced therapies. It's incredible.
So it feels like we're cracking the code for blood cancers. But now, we're staring down an even bigger beast. The next great frontier for this entire technology is solid tumors. We're talking about the cancers that form into masses, things like lung, breast, or brain cancer. And so far, they've been almost impossible to treat with CAR T therapy.
And just to give you a sense of the scale of this problem, 90%. 90% of all cancer cases are solid tumors. So solving this isn't just the next step, it's the key to making this therapy relevant for almost everyone.
So why are they so tough? Well, the best way to think about a solid tumor is like a fortress. It's got physical walls that are tough for T cells to even get inside of. But even if they do, the whole area around the tumor, what scientists call the tumor microenvironment, is just incredibly hostile. It's basically pumping out signals that are designed to shut down and switch off any immune cells that dare to show up.
So if the tumor is a fortress, you can't just send in a regular soldier. You need a commando, and that's exactly what's happening. Scientists are now using CRISPR to build armored T cells, applying advanced tactics to engineer a soldier that can survive and win inside that fortress.
So here's the first strategy, and it's brilliant. You can call it the localized airstrike. Researchers have engineered these T cells to act like tiny little factories. Once they get to the tumor, they start pumping out a super powerful immune activator called IL-12. Now the key is, this payload is only delivered right at the target. This avoids the terrible side effects you'd get if you just flooded the whole body with it. And in early models, this has successfully shrunk tumors that were resistant to everything else. Okay, strategy 2 is all about destroying the supply lines. So instead of just going after the cancer cells, these T cells are engineered to attack a protein that's on both the tumor and its surrounding support structure. You take out the tumor's foundation, and the whole fortress can crumble. But what's truly mind-blowing here is that these cells showed they could cross the blood-brain barrier, which could open up a whole new way to treat devastating brain cancers like glioblastoma.
And you have to understand, this science is moving at an unbelievable speed. What we're talking about right now, it isn't science fiction for some distant future. This is the science that's on the horizon, moving from the lab into the clinic as we speak.
So just how close are we? Well, today the preclinical models are showing amazing results. That's leading to new human trials for solid tumors that are kicking off as soon as 2025. And looking beyond that, the next wave will be even more heavily engineered cells targeting specific markers on really tough cancers, like liver and renal cancer.
I mean, listen to this question. It sounds like something straight out of a sci-fi movie, doesn't it? The idea of diagnosing a fatal genetic disease in a newborn and then treating them with a therapy designed just for them, all in about half a year? Well, it's not science fiction anymore. It just happened. So let me introduce you to baby KJ. He was born with a fatal metabolic disorder and the clock was ticking, literally from day one. But what happened next was just this incredible sprint of science and regulation. A team designed a custom gene therapy, they made it, and they got it cleared by the FDA for treatment. The whole thing, from his birth to his first dose, happened in just 7 months. Absolutely astonishing. So how on earth did they pull that off? Well, the therapy used this super precise tool called a base editor. The best way to think about it is like a genetic pencil that can go in, erase, and rewrite just a single letter of DNA. This pencil was then packaged up inside a tiny bubble of fat, what's called a lipid nanoparticle, or LNP, which basically acted as the delivery truck. They infused it into a system and it went straight to the liver cells that needed that genetic fix. And the results? After 3 doses, KJ's condition stabilized. It was a huge, huge success. You know, this kind of trial is called an N of 1 trial, simply because there's only one patient. And it's a total game changer. It proves that we can create these completely custom medicines for people with ultra-rare diseases. But, and this is a big but, as amazing as this is, it also shines a huge spotlight on the next big challenge. See, the therapy worked so well because those LNPs, they just naturally home in on the liver. But what happens when the genetic problem isn't in the liver? Well, trying to target other parts of the body, that's a whole different ballgame. And this is what takes us from that incredible success story to one of the biggest hurdles in genetic medicine today, the delivery problem. For a disease like cystic fibrosis, for example, which is all about the lungs, you can't just inject a therapy into the bloodstream and cross your fingers. Now, you have to get past some of the body's most well-built defenses.
So first, just a quick refresher on CF. It's caused by mutations in just one gene, the CFTR gene. And that one little fault causes the body to produce this thick, sticky mucus that just clogs up the lungs. It makes it really hard to breathe, and it traps all sorts of nasty bacteria. So the goal for a gene therapy seems simple, right? Just go in and fix that gene. Yeah, getting there is the hard part. You really have to think of the lung as a fortress. It has evolved for millions of years to keep invaders out. First, any therapy you send in has to somehow punch through that thick layer of mucus. Then, if it gets through that, it has to squeeze past these tightly locked gates between the cells. And finally, and this is the most important part, for a cure to actually last, it can't just treat the cells on the surface. It has to go deep and reach the basal stem cells that are constantly regenerating the lung's lining. And right there, that's the core of the problem. If you only manage to edit the surface cells, all you've really done is create a temporary patch, a band-aid at best, because those cells are going to naturally die off and get shed within a few months, and then the patient is right back where they started. A real, durable cure means you have to get to the source, the stem cells. Okay, so how are scientists trying to breach this fortress? They're basically building a whole toolkit of delivery vehicles, and each one has its own set of strengths and weaknesses. The two main approaches they're looking at are based on two totally different things: viruses and synthetic fats. So on one side, you've got viral vectors, things like adeno-associated viruses, or AAVs. I mean, these guys are nature's pros at getting into cells. That's what they do. But they're tiny, and there's a really strict limit on how much genetic cargo you can pack inside. Then on the other side, you've got those lipid nanoparticles, the same LNPs from baby KJ's story. They're generally safer and they can carry bigger payloads, but their default destination is the liver, which, doesn't really help us if we need to get to the lungs. And this is where the AAV approach just hits a brick wall. Look at this number, 4.7 kilobases. That is the absolute maximum amount of genetic code you can cram inside a standard AAV. The problem is just simple math. The healthy CFTR gene by itself is already 4.5 kilobases. Now try to add the CRISPR editing machinery to that, and you're out of room. It literally doesn't fit. So to try and get around this size limit, scientists are testing a really clever workaround, the split vector approach. Basically, they chop up the gene editing machinery and put the pieces into two different viruses. Now, for the therapy to work, both of those viruses have to infect the exact same cell to deliver the complete package. It's a tricky numbers game for sure, but it's a really promising idea. But what if you could just sidestep the lung's fortress entirely? Well, there's another strategy that does exactly that. The idea is to harvest the patient's own airway stem cells, take them to the lab, and then edit them right there. This is a process called ex vivo editing. Once they're fixed, you transplant the corrected cells back into the patient. And the results they're getting in the lab are pretty stunning. Researchers have already managed to restore 70% of normal function in human cells using this method. The big multi-million-dollar question that's still left, though, is how do you get those corrected cells to successfully reimplant and start growing back inside the lung? That's the next big hurdle. Look, the fortress of the lungs is really just one example. This delivery challenge is the central problem for the entire field of gene editing. Every single target organ, whether it's for metabolic disorders or diseases of the brain, it presents its own unique fortress that has to be overcome. It's forcing scientists to get incredibly creative. But here's where it gets really hopeful. For some of these conditions, you don't actually need a 100% perfect fix. Take late-onset Tay-Sachs disease. The data suggests that if you can just restore 10%, just 10% of a critical enzyme's function, that could be enough to stop the neurodegeneration. Think about that. That small of a correction could be completely life-changing. Now let's talk about devastating brain disorders like Huntington's disease. Here, the fortress is maybe the most famous one in all of biology, the blood-brain barrier. It's this incredibly selective membrane that protects our brain from toxins, which is great, but it also blocks pretty much any medicine from getting in. So to get past this barrier, scientists are getting radical. I mean, this is one of the most cutting-edge things happening in medicine. They're combining physical force with genetic engineering. They use focused ultrasound waves, literally sound, to gently and importantly, temporarily open a tiny window in the blood-brain barrier. This creates just enough of an opening for CRISPR-loaded nanoparticles to slip through and get to work. It's incredible stuff. And all of this brings us right back to where we started. All these amazing efforts, breaching the lungs, targeting the brain, They are all part of this new age of medicine, this N of 1 revolution that baby KJ's story really kicked into high gear. And this right here gets to the absolute heart of it. The magic of CRISPR technology is its programmability. It's like software for our genes. It can be rapidly recoded to target any gene you want. And that unlocks the ability to quickly develop these bespoke, custom-made medicines for ultra-rare diseases, the kinds of conditions that affect so few people that they've often been completely ignored by traditional drug companies. This is what we mean by hyper-personalized medicine. It is a fundamental shift from one-size-fits-all drugs to a therapy that is built from the ground up to correct one single person's specific genetic code. It's a completely different way of thinking about disease and how we treat it. Now, nobody is pretending this is easy. The delivery challenges, as we've just talked about, are immense. But here's the thing. For the first time ever, the path forward isn't just some hopeful dream. It's a series of concrete, solvable engineering problems. And that path is getting clearer and clearer every single day. And that brings us right to the single biggest question that frames this entire revolution. When we use CRISPR to change our biology, are we just, offering therapy for diseases? Or are we stepping into the much trickier world of human enhancement? Because that line, it is getting incredibly blurry. As CRISPR gets better and better at fixing what's broken, it's also getting the power to push our biology way beyond its natural baseline. All of a sudden, telling the difference between fixing a problem and adding an upgrade is getting a whole lot harder. So let's kick things off with the revolution happening inside the human body itself. Right on that frontier where treatment starts to look an awful lot like enhancement.
And you really can't talk about this stuff without bringing up the infamous case of He Jiankui. Back in 2018, he just completely shocked the world. He edited human embryos to try and create HIV resistance. Well, fast forward to today, and he's back, now researching other diseases in embryos. His story has really become this powerful cautionary tale about the ethics of it all.
And this is what makes the whole conversation so complicated. I mean, despite being condemned by pretty much everyone, his recent statement just goes to show that the debate around these tools, it's far from over. It's forcing us all to confront these really tough questions right now. This brings up a super important distinction we have to make. What He Jiankui did was germline editing. That means changing the code for all future generations. It's widely condemned. But then there's somatic editing, which only affects the patient and doesn't get passed on. And that's where we're seeing these unbelievable advances in treating diseases. And here is that blurring line in action. Check out these somatic therapies. Knocking out the PCSK9 gene to permanently drop your cholesterol to almost superhuman levels. Or knocking out the CCR5 gene to potentially cure HIV. I mean, yeah, they're treatments, but they also give the body this kind of supernormal resistance. They are, for all intents and purposes, a form of biological enhancement. Which leads us to the next, absolutely wild frontier. Thinking about aging itself not as something that's inevitable, but as a biological process that maybe we can actually hack. Okay, one of the most mind-bending ideas here is something called epigenetic reprogramming. Just think of it like a cellular reset button. Scientists can use CRISPR to go into an old cell and briefly turn on a special set of genes. They're called Yamanaka factors. It doesn't actually change the DNA, but what it does is reset the cell's epigenetic clock, basically bringing back its youthful patterns. It's incredible. So another huge target in this whole anti-aging fight are these things called senescent cells. But a much better way to think of them is as zombie cells. Seriously, they stop dividing like they're supposed to, but they don't die. They just hang around in our bodies as we get older, pumping out all this inflammatory junk that basically drives the aging process. So the goal now is to build these tiny cellular assassins. Scientists are literally designing CRISPR systems that can be programmed to hunt down those specific zombie cells, lock onto their unique genetic signature, and then basically tell them to self-destruct. You clear them out, and you could seriously improve a person's health span. All right, so we've looked at the revolution happening inside our bodies. Now let's pivot to the revolution that's happening all around us, in our food and on our farms. Now, before we go any further, there's a key difference here that you have to understand. Traditional GMOs, right, they often involve taking DNA from one species and sticking it into another. CRISPR's different. Usually, it just makes a tiny snip, a little deletion or small tweak to the DNA that's already there. It's a change that honestly can happen all on its own in nature, and that difference is absolutely critical for what comes next.
And the applications are just amazing. Let's talk about chocolate. Cacao plants are getting hammered by disease. But with CRISPR, scientists just went in and deleted a single gene that was basically holding back the plant's own immune system. The result? A super-resistant plant. No foreign DNA needed. Or how about this? Gluten-free wheat that's safe for people with celiac disease. This is a massive challenge. It means editing 30 to 40 copies of the gluten genes all at once. It's a huge undertaking, and it just shows you the incredible power and precision of this technology. And this isn't science fiction, this is happening right now. In Japan, you can go to the store and buy CRISPR-edited tomatoes that are packed with a compound called GABA, which helps lower blood pressure. It's a perfect real-world example of a functional food made possible by gene editing. And there's a lot more on the way. We're talking about taking the bitterness out of leafy greens to make them taste better, or developing things like seedless berries, which, yeah, people like, but it also helps cut down on food waste. Now, all this innovation has been happening at, well, different speeds in different parts of the world, mostly because of regulations. But now, especially in Europe, the rules of the game are about to change in a big, big way. Put this year on your calendar, 2025, because that's when a major regulatory shift is happening. The European Union is rolling out a brand new mandate for what they're calling New Genomic Techniques, or NGTs. So here's how it works. The EU is creating a two-tiered system. You've got NGT1 plants. These are the ones with edits that could have happened naturally. They're going to be treated just like any conventional crop. No crazy rules, no special labels. But NGT2 plants, the ones with more complex edits, they'll still have to follow the old, much stricter GMO regulations. And this is a total game changer. By creating this faster, friendlier path to market for most CRISPR-edited crops, this decision is going to unleash a massive wave of agricultural innovation in Europe. It brings them much more in line with how things are done in the US and Asia. So when you pull back, we've seen how this one technology is sparking these twin revolutions, one inside our bodies and another on our farms. We really are standing at the dawn of an era where we can actively edit the future of biology itself.
But this incredible, world-changing power comes with a really profound challenge. Who actually gets to use it?
So let's just lay it all out there. This is the central conflict, right? On one hand, you've got the power to cure diseases we once thought were a life sentence. But on the other hand, there's this very real risk that these cures will be so expensive, they'll only be for the super rich, making inequality even worse. This is the tightrope we're walking.
This scientific miracle is running smack into a wall of economic reality. For all its genius, the way we currently deliver this cure creates a massive, massive barrier. And that brings us to a really staggering question, which can be summed up in one number.
Get this, $2.2 million. That is the list price for Casgevy, a brand new CRISPR therapy. And that's not for a whole hospital. That's for a single patient. That's the price tag on a cure right now.
So you gotta ask, why so much? Well, it's because you're not just paying for a vial of medicine. You're paying for this incredibly complex and difficult process called the ex vivo model. Doctors have to literally take a patient's cells out of their body, ship them to a super specialized lab, edit them, and then put them back in. The whole thing involves intense chemotherapy, long hospital stays. It can only be done in a few places in the world. It's just not scalable.
And here's where it gets really tough. This chart shows us the brutal irony of the situation. The people who need this cure the most, for example, for sickle cell disease, are overwhelmingly in places like sub-Saharan Africa or in marginalized communities right here in the US. They're the ones who are least likely to have access to a multi-million-dollar treatment. It's a perfect storm of need meeting an impossible barrier.
And look, this isn't just a problem for individual families. This price tag sends shockwaves through our entire healthcare system. Imagine you're running a state Medicaid program. That huge upfront cost creates what they call a liquidity crisis. It's like your entire budget gets vaporized by just a handful of patients. How is any system supposed to handle that?
But okay, here's the good news. The same brilliant science that got us into this mess is also working like crazy to get us out of it. So let's talk about the path forward, because researchers are basically trying to innovate their way out of this whole economic crisis.
And this slide just lays it out perfectly. The future is all about shifting from that complex ex vivo model, taking cells out, to an in vivo model, which means within the living. The goal? A simple injection that goes into your body and fixes the cells right where they are. This is the absolute game changer for making this technology available to everyone.
The dream is a single-shot cure. You'd walk into a local clinic, get an injection. In a tiny little delivery vehicle, it could be a harmless virus or a little fat bubble called a lipid nanoparticle, kind of like the ones in the mRNA vaccines. It acts like a tiny biological GPS. It finds the exact cells it needs to and makes the edit right there inside your body. No hospital stay, no chemo, no specialized centers.
And I want to be clear, this is not just some fantasy. This is happening. Look at the money pouring into this research. Huge players like the Bill and Melinda Gates Foundation and the National Institutes of Health are investing hundreds of millions of dollars to make these in vivo therapies a reality, specifically targeting diseases that affect the world's most vulnerable people.
So when you think about it, this leap from a $2 million procedure to a potential single shot is about so much more than just science. It's pushing us to a point where we, as a society, have to make some really big, really profound choices.
Because here's the fundamental issue. Our science is just moving at lightning speed. It's incredible. But our rules, our ethical guidelines, our economic models, they just can't keep up. It's like we're building this amazing high-speed bullet train, but we're trying to run it on old, crumbling stone roads. Someone's got to give.
Which brings us to this fork in the road. It's a pretty stark choice, isn't it? Is CRISPR going to be another luxury item that makes the gap between the rich and poor even wider, creating a sort of genetic divide? Or can we make it an instrument of human liberation, freeing all of humanity from these diseases? The decisions we all make in the next few years will push us down one of these two paths.
Okay, to really get a handle on CRISPR, picture this. Our DNA, our entire genome, is like this vast, dark, totally uncharted territory. Now imagine you have a super-powered GPS, or this incredibly powerful searchlight. That's CRISPR. It has the ability to scan through billions, with a B, billions of letters of genetic code to find one single precise location. And what does it do when it finds that exact spot? It makes a cut. It's as simple as that. And this is exactly why you always hear CRISPR-Cas9 being called molecular scissors. It's a system we can program to find and snip DNA with just incredible accuracy. But here's the thing. We didn't invent it. So where did it come from?
Well, it wasn't dreamed up in a high-tech lab. Not a first, anyway. No, this tool was forged in a silent, molecular war that's been going on for billions of years. I'm talking about the endless battle between bacteria and the viruses that hunt them. This tool is their ancient, elegant, and just ruthlessly efficient defense system. But our story doesn't actually start with that epic war. It starts way more quietly, with a little scientific puzzle that popped up way back in 1987. And honestly, it was a complete and total mystery. Right, so over in Japan, a scientist named Yoshizumi Ishino was studying plain old E. coli bacteria, and he noticed something, well, bizarre in its DNA. There were these strange, repeating sequences, and they were separated by these unique little spacer sequences. It looked for all the world like a genetic stutter, and back then, nobody had a clue what it meant. It was just a weird anomaly. And you know what? For a long time, it stayed that way. This timeline really shows you just how long that mystery lasted. For almost 2 decades, these weird repeats, which eventually got the name CRISPR, were basically just a scientific footnote. Yeah, scientists kept finding them in more and more microbes, but their purpose? Total black box. Then, in 2005, the first major clue finally dropped. And it didn't come from a petri dish, it came from the world of computer analysis. Bioinformatics was about to crack this case wide open. So researchers like Francisco Mojica took those unique spacer sequences and ran them through these massive DNA databases. And what they found was, well, it was astonishing. The spacers weren't random noise at all. They were perfect one-to-one matches to the DNA of viruses, the kind that attack bacteria. This led to this incredible, game-changing hypothesis. Bacteria were literally capturing snippets of viral DNA and storing them. It was a genetic memory bank, an adaptive immune system. A brilliant hypothesis, right? But it's just a hypothesis until you can prove it. And that proof would come in 2007. And believe it or not, it was all thanks to a very practical problem that was causing headaches for the dairy industry. So get this, scientists at the food company Danisco, the yogurt people,
They were just trying to protect their yogurt-making bacteria from getting wiped out by viruses. And they watched, in real time, as bacteria that survived an infection incorporated brand new spacers into their DNA. And where did those spacers come from? They were taken directly from the virus that had just attacked them. It was the smoking gun, the first direct proof that CRISPR was basically a bacterial vaccination card written right into their own DNA. Okay, so the what was now crystal clear, but the how was still a mystery. I mean, how did this genetic memory bank actually stop a virus in its tracks? The race was on to figure out the machinery behind it. And what scientists found was a system with a few key parts. First up, you've got the CRISPR array. That's the memory bank where all the viral DNA is stored. Second, you've got the Cas proteins. Those are the actual scissors that do the cutting. Third, to guide those scissors to the right spot, the system makes a little copy of the viral memory, a molecule called CRISPR. And then, the final piece of the puzzle, discovered in 2011 by Emmanuel Charpentier's team, a helper molecule called tracRNA. This little helper turned out to be absolutely crucial for getting the Cas9 protein ready for action, kind of like unlocking the scissors so they're ready to cut. And the discovery of that helper RNA, it set the stage for the absolute pivotal moment in 2012. This is when this natural defense system was about to be transformed from, a biological curiosity into a truly world-changing technology. So here it is, the moment of pure genius. Jennifer Doudna and Emmanuel Charpentier realized they could dramatically simplify the whole system. They figured out how to fuse the guide RNA and that helper RNA into one single elegant molecule. This masterstroke turned a complex three-part natural system into this incredibly simple, programmable tool. Now, all you had to do was give the Cas9 protein one single guide for whatever location you wanted, and it would go there and cut. It became a true plug-and-play device for editing genomes. And things moved fast. The very next year, researchers like Feng Zhang and George Church took that final massive leap. They showed that this amazing bacterial system could be adapted to work inside the much, much more complex environment of our own cells, human cells, and other animal cells. The era of precision gene editing had officially begun.
Well, before it ever saw the inside of a lab, it was an ancient microscopic weapon.
How does a single cell, a thing with no brain, no nervous system, how does it remember its enemies? How does it learn to spot a predator it's already fought and know how to fight back?
You gotta understand, for a bacterium, life is basically a constant state of war. They are under relentless attack by viruses. We call them bacteriophages, that are always trying to inject their own DNA and turn the cell into a mindless virus copying factory. It is a microscopic arms race that's been raging for billions of years.
So how do they actually fight back? Well, that's what we're here to break down. We're going to start on the viral battlefield itself. Then we'll get into the nitty-gritty of gathering intelligence. From there, we'll see how they build a guided weapon, face what I'm calling the assassin's paradox, and finally, figure out the most important thing, how to tell friend from foe.
Okay, so to survive this non-stop viral attack, bacteria had to evolve something really, really clever. A sophisticated, adaptive immune system. And I bet you can guess its name. Yep, it's CRISPR.
So what is CRISPR really? At its heart, it's like a genetic scrapbook of all the viruses that have ever attacked it. The bacterium literally snips out a tiny piece of the invader's DNA, saves it, and then uses that memory to hunt down and obliterate that same virus if it ever dares to show its face again.
And this whole defense system works in three pretty logical stages. First up is adaptation. That's all about getting the memory. Next is expression, which is where they actually build the weapon. And finally, you have interference, time to use that weapon and take out the target.
All right, let's dive into stage one, adaptation. You can think of this as a full-on counterintelligence mission. The goal? Capture a piece of the enemy's blueprint.
The main spies in this operation are a pair of proteins, Cas9 and Cas2. They team up to form this little complex that acts just like a molecular ruler. Its whole job is to find the invader's DNA, measure out a perfect little snippet, and snag it as a souvenir.
And this whole thing is incredibly precise. First, that Cas1, Cas2 complex gets a hold of the viral DNA. Then it measures out and snips off a tiny fragment of a very specific length. We're talking about 30 base pairs, which is like 30 rungs on the DNA ladder. This little piece they capture is called a prespacer, but you can just think of it as a viral mugshot. And then in the final step, this mugshot gets woven right into a special section of the bacterium's own DNA, a place called the CRISPR array.
It's basically a genetic most wanted gallery. Now, to make sure this whole capture is super accurate, some bacteria have an extra helper protein called CSN2. And what it does is pretty cool. It forms a ring around the DNA, kind of like a clamp you'd use in woodworking, just to hold everything perfectly still so they get a clean cut and a perfect integration.
Okay, so now our bacterium has its most wanted gallery full of viral mugshots. But hey, a photo album of bad guys is pretty useless if you can't do anything with it, right? And that brings us to stage 2, expression. This is where we turn that memory into an actual guided weapon.
So how do they do it? Well, first, the cell makes a copy of the entire CRISPR array, that whole library of mugshots. But this copy isn't DNA, it's a slightly different molecule called RNA. And it comes out as this one long streamer that has all the different enemy profiles on it, one after another. This long RNA streamer then gets met by a couple of other key players. First, another little bit of RNA called a tracer comes in and latches on, basically acting like a dotted line that says cut here. Then, an enzyme called RNase 3 shows up, and it's basically a pair of molecular scissors. It just goes along and chops that long strip at every single mark, turning it into a bunch of individual weaponized guides.
So each one of those little chopped up pieces is now a guide RNA. This guide, our viral mugshot, gets loaded into another protein, and this is the one you've probably heard of, the famous Cas9. So if you think of Cas9 as the assassin, the guide RNA is the intel, the picture that tells it who to target. And together, they form the ultimate search and destroy team.
And this, my friends, brings us to the climax, the interference stage. The bacterium is armed and dangerous. The Cas9 assassin is on the loose, guided by its RNA mugshot, patrolling the cell, just looking for a match. But this creates a huge, potentially fatal problem. A paradox, really.
See, this complex is ridiculously good at its job. It just drifts through the cell, constantly scanning every piece of DNA it bumps into. And the moment it finds a sequence that is a perfect match for the guide RNA it's holding, it latches on and gets ready to cut.
But wait a second, think about it. Where did that guide RNA come from? It's a copy of a sequence that's stored in the bacterium's own DNA. The cell is literally carrying the blueprint for its own destruction. So what stops the Cas9 assassin from turning on its creator and committing cellular suicide? Why doesn't it just shred its own genome?
The solution is, well, it's just so simple and elegant. It all comes down to a tiny, critical piece of information. You can think of it like a secret password that lets the system tell the difference between enemy and me. This password is a very short DNA sequence called a PAM. That stands for Protospacer Adjacent Motif. Now here's the key. The PAM sequence has to be sitting right next to the target sequence, but only on the invader's DNA. And Cas9 is programmed with one simple, unbreakable rule. If there's no PAM, there's no cut, period. It absolutely will not slice any DNA unless that password is there. And this is how it all works. So, on the invading virus DNA, you've got the target sequence, that's the protospacer, and right next to it, you've got the PAM password. Cas9 sees both, and boom, it cuts. But if you look at the bacterium's own genetic archive, it has the matching sequence, the spacer, but it's next to a different bit of DNA that does not have that PAM sequence. No password, no cut. The bacterium is totally safe. It really is that simple. It's like binary code, pure digital logic. PAM present? Destroy. PAM absent? Ignore. This fundamental yes or no switch is the one and only safeguard that keeps the bacterium from destroying itself with its own super powerful weapon. So when you step back and look at the whole thing, it's just an unbelievably elegant system, isn't it? Honed and perfected over millions of years. You've got a way to create a memory, a way to build a targeted weapon based on that memory, and then a simple yet totally foolproof safety switch to keep everything in check.
We are not talking about some passive, static little rock floating around. No way. Cas9 is an active, dynamic piece of biological hardware that literally changes its shape to get the job done. And understanding that movement, that's the real key to understanding how gene editing actually works on a molecular level. So, here's our game plan. We'll kick things off with why this dynamic machine idea is so important. Then we'll look at its basic two-part blueprint. From there, we'll break down the three essential components: the GPS, the scissors, and of course, the software that tells the whole thing where to go. Okay, first things first. Let's really lock in this idea of Cas9 as a machine that's constantly in motion. And this is the absolute core concept you've got to grasp. This enzyme doesn't just sort of bump into DNA and snip it. It has to twist, turn, open up, and clamp down in a really precise sequence. We're talking about massive physical changes that allow it to first find, then verify, and only then cut its target. You can think of it like this intricate molecular dance, and every single step has to be perfect for it to work.
So how is a machine like this built? Well, at its core, it has this beautifully simple but incredibly powerful two-part design.
The fancy term is bilobed architecture, but don't let that intimidate you. It just means the protein is made of two main sections, or lobes. And these lobes are connected, but they have totally different jobs. This separation of duties is what makes the whole thing work. It's kind of like a high-tech pair of pliers. One handle is for aiming, and the other one is for cutting.
And this table just lays it out perfectly. On one side, you've got the recognition lobe, or rec lobe for short. This is the machine's GPS interface. Its whole purpose is to scan the genome and find the one exact DNA sequence we're looking for. Then on the other side, you have the nucleus lobe, or nuke lobe. This is the heavy machinery. This is the part that actually holds the tools to cut the DNA.
Now, what connects these two lobes is just as brilliant. It's a little structure called the bridge helix, and it basically acts like a hinge. See, when the guide RNA, that's the software we'll get to in a minute, is loaded in, this hinge physically shifts, and that shift creates a positively charged channel right down the middle of the machine, which is just perfect for grabbing onto the negatively charged DNA strand. It's like a magnet.
Okay, let's zoom in a bit on that first part. Let's really look at the GPS system of the Cas9 machine. So the rec lobe itself isn't just one solid piece. It's actually made of three smaller subcomponents called domains. Rec 1 is the biggest, and you can think of it as the main anchor point for the guide RNA. It holds everything in place. Then you've got Rec 2 and Rec 3. These guys are much more flexible, and their job is mission critical. They are the proofreaders. They double and triple check that the guide RNA has found the exact right spot before the machine is cleared to make a cut. I absolutely love this metaphor from the source material. It's just so vivid. If the guide RNA is the programmable searchlight that's scanning the genome for the target, then the Rec1 domain is what firmly holds the handle. It keeps that searchlight stable and pointed exactly where it needs to be. It's a fantastic way to picture what's happening.
Okay, so the GPS has locked onto its target, it's given the all clear, now it's time for the other half of the machine to go to work, the nuclease lobe, aka the scissors. And here's something that makes Cas9 really special, something that sets it apart from a lot of other DNA cutting enzymes. Instead of having just one cutting site, or needing two separate proteins to come together, Cas9 has two different cutting tools built right into its own single protein. This design gives it an incredible amount of precision and control. So what are these two tools? They're called the HNH and the RUV-C domains. Each one has a very specific assignment. The HNH domain is super flexible. It just kind of waits around until the target is 100% confirmed, and then it swings into position to cut the target strand. That's the side of the DNA ladder that perfectly matches the guide RNA. At the same time, the RUV-C domain gets to work on the other side, the non-target strand, using a really precise chemical reaction to make its cut. One tool for each side of the DNA. It's beautifully efficient. So we have the GPS, we have the scissors, but how does this machine know where in the 3 billion letters of the genome to even go? Well, that brings us to the final and maybe the most revolutionary component of all, the software. The software is this amazing engineered molecule called a single guide RNA, or sgRNA for short. See, in nature, bacteria actually use two separate RNA molecules to get this done. The massive breakthrough for gene editing was figuring out how to fuse those two into one elegant programmable instruction set that we can just design on a computer and build in a lab. This little piece of software has two main parts. The first 20 or so letters, the nucleotides, are called the spacer. This is the part that scientists can customize to match any DNA sequence they want to find. This is the beam of our searchlight. Then you have the rest of the molecule, which is called the scaffold. And this is the structural handle that the RSC1 domain physically grabs onto. It folds into this very specific shape that plugs right into the Cas9 protein like a key into a lock.
And you have to understand, this part is absolutely essential.
Without that scaffold handle plugging in, the Cas9 protein is just a useless, inactive lump of protein. It can't do anything. It's the scaffold that flips the power switch, causing that hinge to shift and opening up the channel for the DNA to come in. The hardware, no matter how powerful, is totally useless without the software.
So let's put it all together. Let's watch the machine run step by step. First, the software gets loaded. The sgRNA binds to Cas9 and activates it. Second, the target is acquired. The rec lobe's searchlight scans the DNA and finds a match. Third, the match is verified. The proofreading domains double-check for a perfect fit. And finally, only after everything is confirmed, the NUC lobes, 2 scissors, H&H, and Roof C swing into action and make a clean, precise cut. We're going to break down how the CRISPR-Cas9 system pulls off a feat of molecular detective work that is frankly almost impossible to believe. So let's get into it. All right, to really wrap your head around the challenge we're talking about, imagine this. I give you a mission. Find one specific single word somewhere inside a library that has 500,000 books, and each of those books is thousands of pages long. I mean, where would you even start? Well, that's pretty much the exact problem our little molecular agent Cas9 faces every single time it goes to work. See, the library it's searching through isn't made of paper and ink, it's the human genome. And that library has over 3.2 billion letters of genetic code. It is just a staggering amount of information to sift through. And the word it's looking for? It's a tiny little sequence, usually just about 20 letters long. So the mission is to find that one specific 20-letter sequence inside a 3.2 billion letter book. How on earth does it pull that off? This brings us to what really seems like an impossible search. Now, you might be thinking Cas9 just reads the entire genome, letter by letter. But that's not how this spy operates at all. Just think about it. The energy it would take for a cell to unzip and read all 3.2 billion of those letters, it's just not feasible. So brute force is off the table. Instead, Cas9 uses this incredibly elegant strategy, a whole series of checkpoints that lets it find its target with unbelievable speed and accuracy. And the success of this whole mission really hinges on finding one specific clue first. This is the system's most important filter, and it's called the PAM signal. So, this signal is called the PAM, which stands for Protospacer Adjacent Motif. The easiest way to think about it is like a little 3-letter signpost, specifically NGG, that's scattered all throughout the genome. As Cas9 is just randomly bouncing around inside the cell nucleus, it's not actually looking for your 20-letter target. No, it's only looking for this 3-letter signpost. That's the crucial part. It ignores almost everything else. And what's so cool here is how physical this all is. The Cas9 protein has a part called the PI domain, and it uses what you can basically think of as 2 probing fingers. These are actually arginine molecules that literally reach down into the groove of the DNA helix, and they're feeling around for the specific chemical bonds that tell them, hey, this is a PAM sequence. And this process shows you just how efficient the system is. Cas9 bumps into a random piece of DNA, the fingers check for the PAM. If it's not there, the whole thing just detaches in milliseconds and continues its random walk. But if it does find a PAM, it stops, it pauses, latches onto that spot, and only then does it decide to investigate further. This super simple filter just massively cuts down the search area.
Okay, so our agent, Cas9, has found a PAM signal and it's latched on. But the target sequence is still locked up tight inside that DNA double helix. So that brings us to the next phase, prying open the code.
For years, scientists figured that the Cas9 protein just used brute force, that it physically bent and ripped the DNA strands apart. But as it turns out, the new research shows it's a much more subtle, cooperative attack. It's not just the protein working alone. The guide RNA itself plays a super important active role in cracking open that helix. It's really this beautiful 2-step mechanism. First, the Cas9 protein grabs the DNA's backbone and creates this little kink, something called a phosphate lock, which basically makes the structure unstable right at that spot. Then, the guide RNA takes advantage of that weakness and physically wedges its first letter, its first nucleotide, right into that unstable spot. It's perfect teamwork. The protein picks the lock, and the RNA pushes the door open. All right, the DNA is unzipped. The moment of truth has arrived. The guide RNA now has to perform this high-stakes handshake with one of the DNA strands to see if it's the right password. When that guide RNA finds its matching sequence on the DNA, they bind together and they form this special structure called an R loop.
This is the physical proof that we have a match. You literally get a new hybrid molecule made of RNA and DNA, and the other DNA strand just gets pushed out of the way, left hanging.
And this check isn't random at all. It's incredibly methodical. It's directional. It starts right next to where the PAM was and zips up, base by base, comparing the guide RNA to the target DNA. It's like zipping up a jacket, one tooth at a time to make sure everything aligns perfectly.
Now, this is probably the most critical step of all. The system has this extremely strict security check right at the beginning of the zipper. This initial section, about 8 to 10 letters long, is known as the seed region, and it demands a perfect 100% match, no exceptions.
So if there is even one single mistake, one mismatch in this seed checkpoint, the whole handshake fails. That R loop just collapses and the mission is aborted. Cas9 lets go of the DNA and goes back to searching somewhere else. This checkpoint is the main reason why Cas9 is so incredibly specific and doesn't just go around cutting up random DNA.
So, you'd think we're done, right? We found the PAM, we opened the DNA, we confirmed a perfect match in the seed region. But believe it or not, even that is not enough to make the cut. There is one final safety switch that has to be flipped before the molecular scissors get activated. This is a brilliant built-in safety feature. It's a confirmational checkpoint. You can think of it like the system asking, "Are you absolutely sure?" before it does anything permanent. Just because you found the target doesn't mean you should automatically fire the weapon.
And this final sequence is just pure molecular drama. Once the full 20-letter handshake is confirmed, an internal go signal is sent. This triggers a massive change in the protein's shape. A piece of it, called the HNH domain, which is the actual cutting blade, physically swings a huge distance, 25 angstroms, to lock into its cutting position. Only then, once it's locked and loaded, are both strands of the DNA finally cut.
This whole system, with all its layers of security, is just incredible. But it does raise a really important question. What happens if the match is pretty good, the R loop forms, but for some reason, maybe a few mismatches way at the end of the sequence, that final go signal is never sent? What if that final switch doesn't flip? And this brings us to a really key insight into how this works in the real world. If that final switch fails, Cas9 will bind to the DNA, and bind to it tightly, but it won't cut. It just sits there, stuck. And now it's become a roadblock on the DNA that can block other important cellular machines from doing their jobs. This binding without cutting is actually a major source of what scientists call off-target effects, and it's one of the biggest challenges they're working to solve in gene editing. It just goes to show you that even in a system this close to perfect, the complexities of biology always leave us with another fascinating puzzle to solve.
How do you take a tiny piece of a bacterium's immune system and get it to work inside one of our own incredibly complex human cells? Well, today, we're going to dive into that exact story. It's an incredible engineering journey full of clever hacks and brilliant workarounds that turned CRISPR-Cas9 into the revolutionary tool it is today.
So the whole challenge really boils down to this. We're talking about two completely different worlds, separated by billions of years of evolution. You've got the bacterial cell where the DNA just kind of floats around. Simple. But then you have a human cell where our precious genome is locked away inside a fortress, the nucleus. And that creates the very first and maybe the biggest problem. How in the world do you get your tool to the target?
And the tool we're talking about, Cas9, is this big protein. To a human cell, it is a complete and total foreigner. See, our cells have these really sophisticated systems for deciding what gets in, what gets made, and where everything goes. You can't just toss a bacterial protein into the mix and, hope for the best. It's not going to work. It needs a guide. It needs a pass.
Which brings us to hurdle number 1, getting past that gatekeeper. The nucleus is surrounded by a membrane with these little pores that act like security checkpoints. And Cas9 is huge. It's way too big to just drift on through. It absolutely needs an all-access pass to get to the DNA.
The solution scientists came up with is just so elegant. It's a fantastic piece of bioengineering. What they did was they attached something called a nuclear localization signal, or NLS, right onto the Cas9 protein. The best way to think of it is like a shipping label, like on an Amazon package. It's a short tag that basically tells the cell's internal machinery, hey, this package right here, this belongs in the nucleus.
So here's how it works. Once that NLS shipping label is on there, these specialized proteins inside the cell, they're called importins. They see it and they recognize it. They grab onto the Cas9 and act like a personal escort, actively pulling the whole thing through the nuclear pore.
So it's not about breaking down the door. It's about having the right key card so the security guards let you right in. And just like that, problem 1 solved. But okay, getting the tool inside the nucleus is only half the battle. What if you want the cell to build the tool for you? Well, that means you have to deliver the gene for Cas9, and this creates a whole new problem, a language barrier. You see, while the basic genetic code is pretty much universal, different organisms have different, let's call them preferences for certain DNA words. The bacterium where Cas9 comes from, Streptococcus pyogenes, speaks a genetic dialect that's really rich in A and T bases. Human cells, on the other hand, speak a dialect that's rich in G and C. So if you just stick the bacterial gene into a human cell, the cell's machinery kind of stalls. It's almost like it's struggling to understand a really thick, unfamiliar accent. So what do? We have to translate. The solution is this process called codon optimization. Scientists literally rewrite the entire gene for Cas9. They swap out all the bacteria-preferred words for the human-preferred ones. But here's the cool part. It doesn't change the final protein at all. You get the exact same tool, but the instruction manual is now written in fluent human, which means the cell can build it super efficiently. Okay, so we've solved location and we've solved language. Now for the logistics. You've got this perfectly engineered tool. How do you actually get it into potentially millions of cells, especially inside a living person? This is the special delivery problem. And it turns out there isn't just one single answer. The method you pick really depends on what you're trying to do, and it always involves this critical trade-off between how effective it is and how safe it is. This table breaks it down nicely. You could deliver the instructions as a circular piece of DNA called a plasmid. The tool gets made for a long time, but because it's hanging around so long, the risk of it making unwanted cuts, what we call off-target effects, is higher. A better way is using mRNA. It's temporary. The cell makes the tool, but then the instructions disappear pretty quickly, which lowers the risk. But the safest method of all is delivering the ribonucleoprotein, or RNP. And this is exactly why RNP is the gold standard for clinical use. You're not even delivering the instructions anymore. You are delivering the fully assembled, ready-to-go tool directly into the cell. It gets in, makes its one specific cut, and then the cell breaks it down within hours. It's a hit-and-run mission, and that gives you the highest precision with the absolute lowest risk. But this leads us to one final and really fascinating problem. For a lot of therapies, especially ones that need to target organs deep inside the body, scientists use a delivery vehicle, usually a harmless engineered virus like AAV. And the thing about these viruses is they have a very, very strict cargo size limit. So the original Cas9, this sCas9, it's a beast. At 1,368 amino acids long, it barely fits inside the delivery truck. This whole packaging problem started a massive hunt for a smaller version. And eventually, researchers found one in a different bacterium, Staphylococcus aureus. And this one, sCas9, is way smaller, only 1,053 amino acids. Finding this smaller version was a genuine breakthrough. I mean, it wasn't just some small improvement, it was the key that unlocked our ability to use these AAV viruses for in-body gene therapy. All of a sudden, the delivery vehicle was plenty big enough for the cargo. And that opened up a whole new world of therapeutic possibilities. Okay, so that just begs the question, right? If this revolutionary technology is just about breaking DNA, where does the actual editing part come from? How does that even work? And here's the beautiful, incredible twist. The real magic isn't in the cut, it's in the cleanup. The edit is what happens when the cell absolutely scrambles to repair the life-threatening damage that CRISPR just caused. The true editors? They were inside the cell all along. So to really understand this, we need to meet the cell's DNA repair crew. There are two main teams that show up when there's a break, and believe me, they have completely different ways of getting the job done.
On one side, you've got the smash and grab crew. Its technical name is NHEJ. It's super fast, kind of messy, and it's the cell's go-to default response. On the other side, you've got the photocopier, or HDR. This one is slow, careful, and incredibly precise, but it's also pretty rare. And as you're about to see, these two are always in a race against each other.
Let's start with NHJ. Think of this pathway as the cell's paramedics. A double-strand break in your DNA is like a 5-alarm fire. It's catastrophic. And this crew's only job is to patch it up as fast as possible to stop the cell from dying. They are not worried about making it look pretty. They are all about survival right now.
So how does this emergency repair actually happen? Well, first, proteins called Ku act like clamps. They just grab onto the two broken ends to keep them from floating away. Then they call in some backup, other proteins that might chew back the ragged ends a little bit. And finally, a molecule called DNA ligase 4 comes in and basically acts like superglue, just sticking the two ends back together. But this rushed patch job, it almost always leaves a scar. And those scars, funnily enough, they're often exactly what scientists are looking for. They're called indels. Basically, a few DNA letters get randomly added or deleted right at the cut site. Now, if this happens inside a gene, it can mess up the instructions so badly that the gene just stops working. And that's what we call a gene knockout. It's the most common thing people do with CRISPR.
But what if your goal isn't just to break a gene, but to actually fix one? Or maybe even add a new one? Well, for that, you need the other crew, HDR, the cell's meticulous architect. This pathway doesn't just slap a patch on the break. It rebuilds it perfectly, using a blueprint.
Now, this is a much more elegant process. First, a set of proteins finds the break. Then, it carefully chews back one of the DNA strands to create this little overhang. A really key protein, RAD51, coats this single strand and uses it like a search bar to find a matching sequence somewhere else in the cell to use as a template. Once it finds it, it perfectly copies the information from that template to repair the break. No mistakes, no scars. So you have this fast and sloppy pathway, and then you have a slow and precise one. This whole situation creates the central problem in gene editing. It's a constant standoff between these two systems inside every single cell. And this chart really just says it all, doesn't it? Look at that difference. In the race to fix that break that CRISPR just made, the fast and messy NHEJ pathway wins almost every single time. The precise HDR pathway, the one we actually need for the really sophisticated edits, it barely gets a look in. So why? Why is it so lopsided? It all comes down to the cell cycle. See, the NHEJ emergency crew is basically on call 24/7 through every single phase of a cell's life. But HDR, our careful architect, it can only work during very specific windows, the S and G2 phases. That's when the cell has already duplicated its DNA, so there's a perfect copy nearby to use as a template. The rest of the time, HDR is totally off the clock. And this brings us to a really crucial and honestly kind of brutal number. When scientists try to do a precise edit, like inserting a new piece of genetic code, which they call a knock-in, the success rate is often painfully low. We're talking 1 to 10% efficiency. It's a direct result of this internal competition, where the pathway we want is just so rarely used by the cell.
Okay, so if the cell is so stubborn, how does scientists ever get those precise edits to work? Well, this is where the real genius comes in. They figured out how to hijack the cell's own machinery and trick it into doing what they want.
To boast that tiny 1 to 10% success rate, researchers don't just add the CRISPR scissors. At the same time, they flood the cell with millions of copies of a custom-built piece of DNA. They call it a donor template. And this little piece of DNA contains the exact genetic change they want to make. The hope is that the cell's HDR machinery will accidentally grab one of these artificial templates to fix the break, and in doing so, write the new code right into the genome.
But that's not the only trick they have. There's another really clever way to get a specific result, and it involves exploiting a little glitch in the system. A third repair pathway that's also error-prone, but in a very specific and very predictable way. The key to this whole thing is something called microhomology. All it means is that sometimes you have these very short, identical sequences of DNA letters on either side of a break, and every now and then the cell gets a little confused and uses these tiny matching spots to stitch the DNA back together. Here's how this little glitch plays out. First, the cell trims the broken ends, and that exposes these little matching sequences. Then it sticks those two matching sequences together. But wait, what about all the DNA that was in between those 2 points? Well, as you can see here, it just gets clipped off. It's thrown away. The result is always the same, a perfect deletion of the entire sequence that was between those two microhomologies.
And this is the beautiful part. Yeah, it's a mistake. It's a cellular glitch. But It's a predictable mistake. So if scientists can find 2 of these little microhomology regions and just tell CRISPR to cut right in between them, they know with almost total certainty that the entire chunk of DNA in the middle will get deleted. They've turned a bug into a feature.
Which brings us right back to where we started. CRISPR itself is just the trigger. It's the hammer that breaks the window. The final result, whether you get a messy scar, a perfect replacement, or a clean deletion, is not decided by the scissors. It's all decided by the cell's internal repair crew and how they choose to clean up the mess you just made.
You know, when we talk about gene editing, one word probably pops into your head: CRISPR. But that's really just chapter 1 of an incredible story. Today, we are diving deep into the quest for the perfect gene editor, a journey that's moved way, way beyond those original molecular scissors. Look, there's no question, CRISPR-Cas9 was a revolution. It gave us the power to edit the very code of life. But it also forced us to ask a really tough question. What if the thing that made it so powerful was also its biggest flaw? What if that first version was just too dangerous to ever really use in medicine? And that gets us right to the heart of the problem with the original CRISPR system. See, its whole magic trick was that it acted like a tiny pair of scissors, cutting straight through a strand of DNA. But as scientists quickly found out, making that cut is an incredibly risky move inside a living cell. The technical term for this is a double-strand break, or DSB, and that's exactly what CRISPR-Cas9 does. It slices through both sides of that DNA ladder. Now, this sends a panic signal to the cell, like a giant flashing red light that says, "Emergency, repair needed now." The problem is, the cell's emergency repair crew, well, it can be a little clumsy. And that clumsiness is where the danger lies. Making these breaks can set off a whole chain reaction of bad things. The cell might just decide to hit the self-destruct button, or it might accidentally delete a huge chunk of genetic code while trying to patch the hole. Worst of all, the broken ends of 2 completely different chromosomes could get stuck together. It was clear they needed a much safer way to do this. So that urgent need for safety led to the very first major upgrade in gene editing technology. The idea was simple, really. It was time to trade in the brute force of those molecular scissors for the delicate precision of a surgeon's scalpel. And the solution was absolutely brilliant. It's called a nickase. By tweaking just a tiny part of the Cas9 protein, scientists basically broke one of its 2 cutting blades. So what you get is an enzyme that doesn't create that dangerous double-strand break anymore. Instead, it just gently nicks a single strand of the DNA. A tiny change, but one with a massive impact on safety. So you might be wondering, how do you make an edit with just a single nick? Well, you use 2 of them. It's a technique called double nicking. You send in 2 of these nickases, each targeting opposite strands really close to each other. This creates a staggered break that the cell can fix way more accurately. But here's the genius part. Let's say one of the nickases goes rogue and nicks the wrong place in the genome, an off-target edit. The cell's repair system just sees a simple, single-strand nick and fixes it perfectly, no harm done. This simple trick basically wiped out off-target mutations. But you know, even a scalpel is still a cutting tool. The next leap forward wasn't just an improvement, it was a complete change in strategy. Scientists started asking a groundbreaking question. What if we could stop cutting DNA entirely? What if we could just rewrite it? And this comparison really shows you how huge that idea was. The old way, the cutting method, meant breaking the DNA and just hoping that the cell's messy repair crew got the fix right. The new way? It skips all that. It uses molecular chemistry to directly change one letter of the DNA code into another, all without making a single dangerous break. This amazing new tech is called base editing. Here's how it works. You take a dead Cas9, it's been modified so it can find a specific DNA sequence, but it can't cut at all, and you glue it to a special enzyme called a deaminase. So the Cas9 part acts like a GPS, taking the whole package to the exact right address in the genome, and then the deaminase enzyme does the actual chemical rewrite. Okay, so let's get into how that rewrite actually works. Once the Cas9 finds its spot and unwinds the DNA, the deaminase enzyme gets to work. One type of base editor can find a cytosine, a C, and chemically transform it into a molecule that the cell reads as a thymine, a T. Another type, which was incredibly difficult to create, does something similar, converting an adenine, or A, into something the cell reads as a guanine, or G. The code is changed, letter by letter, and not a single cut was made. So just how big of a deal is this? Well, it's absolutely staggering.
Scientists estimate that these base editors have the potential to correct up to 60% of all known human genetic diseases that are caused by a single-letter mistake in our DNA. That's thousands of diseases. But even this incredible technology had its limits. You can think of a base editor like a pencil. It's super precise, but it can only make two specific changes, turning a C into a T or an A into a G. It can't swap a C for an A, and it definitely can't add or delete letters. To get that kind of power, science needed one final breakthrough. And that breakthrough finally came. It's called prime editing. And this is the technology that takes us beyond just making simple edits and gives us something that works like a true word processor for the genome itself. Honestly, the scientists who developed it put it perfectly. They said, if Cas9 is scissors and base editors are a pencil, then prime editing is a word processor. It's literally like having a search and replace function for the code of life. So the prime editing system is this really elegant piece of molecular engineering. It actually combines two things we've talked about: the gentle Cas9 nickase with an enzyme called a reverse transcriptase, which has the amazing ability to write new DNA based on an RNA template. And the whole thing is guided by a new, special kind of RNA that carries both the address of where to go and the new genetic text that needs to be written. And the way it works is just beautiful. First, the nickase makes that safe single-strand snip. Then that special guide RNA swoops in and binds right to the nicked strand. Next, the reverse transcriptase gets to work, using the guide RNA as a template to literally write the new, corrected genetic code right on top of the old sequence. Finally, the cell's own repair team comes in, removes the old flap of DNA, and seals the newly written sequence in place. It is a direct search and replace. So let's just pull this all together. We started with CRISPR-Cas9, the powerful but risky scissors. Then we evolved to base editors, the precise pencil for fixing very specific typos. And now we've arrived at prime editing, the word processor that can do it all. Any base-to-base swap, plus add or delete small chunks of code, all with incredible precision. When you step back and look at it, this journey has been one of the fastest, most incredible technological revolutions we've ever seen in science. It's the story of going from a blunt, powerful instrument to a tool of almost unimaginable, exquisite precision. And really, that's the core of the story. In what feels like the blink of an eye, the scientific world took an inherently dangerous idea, cutting our DNA, and step by step, they engineered the risk right out of it, creating safer, more powerful, and more versatile tools with every single innovation. Okay, a really key point here is how the therapy is delivered. On the left, you've got the ex vivo approach, which literally means outside the body. So for a blood disorder like sickle cell, doctors can actually take a patient's cells out, edit them in the lab, and then put them back in. But on the right, you have in vivo, which is inside the body. For something like ATTR amyloidosis, that's a disease caused by a haywire protein in the liver. The CRISPR therapy is just injected right into you, and it finds its way to the target all on its own. It's two totally different strategies for two very different problems. The scissors didn't just open a door. It's more like they revealed this huge, incredible workshop, just packed with new tools that we're only now starting to figure out how to use. The ability to cut DNA, that was the big breakthrough, for sure. But what we can do after the cut, well, that's where the real revolution is happening. This technology isn't just for people. Over in agriculture, CRISPR is making these really subtle but powerful tweaks. Take these mushrooms, for instance. Scientists can use CRISPR to just turn off the one gene that makes them go brown. And here's the key difference. Because no foreign DNA is added, these kinds of edits are often regulated completely differently from what we think of as traditional GMOs. It's just a much, much more precise way of doing things. All right, section 3. Now, what if I told you CRISPR's biggest strength wasn't editing at all, but finding things? What if it could be one of the most powerful search tools on the entire planet? Well, this is where a whole new world of applications opens up: diagnostics. Yeah, it turns out our buddy Cas9 that we always talk about, it's just one member of this huge family. There are other Cas proteins like Cas12 and Cas13, and they have some different and honestly kind of weird properties. Instead of just making one clean cut when they find their target, they react a little differently.
And this shows that weird property perfectly. When these other Cas enzymes find what they're looking for, let's say a piece of the COVID-19 virus, they don't just cut it once. They go into a total frenzy, shredding any genetic material in the area. So scientists have figured out how to harness this little destructive habit. They add tiny reporter molecules that only glow when they get shredded. And the result? You get this incredibly sensitive test on a simple paper strip that tells you if the virus is there. It's genius. Okay, so this brings us right to the core question, doesn't it? We've seen it edit genes, we've seen it turn genes on, we've seen it detect viruses. The whole molecular scissors metaphor is, well, it's starting to fall apart. So what is the real nature of CRISPR? Section 4. This is the idea that changes everything. To really, truly get what this technology is, you need to throw out the scissors metaphor. Just toss it. A much, much better analogy: think of CRISPR as a programmable, universal GPS for the genome. This right here is the key insight. The most important part of CRISPR isn't the cutting, it's the finding. It's a system that you can program to find almost any sequence of DNA in any living thing with just breathtaking precision. And once it gets there, what it actually does is totally up to you. The cutting part? That's just one possible attachment you can give it. The hardware and software analogy on this slide is just perfect. The Cas protein is the hardware. It's the physical homing device that travels along the DNA. It stays the same no matter what the job is. The guide RNA? That's the software. It's the specific GPS coordinates you punch in. So if you want to target a different gene, you don't build a new machine. You just write new code. You just make a new guide RNA. And here's the beautiful payoff of that idea. You can attach all sorts of different tools, different molecular cargo to that GPS. You attach a nuclease, and yep, you get your classic scissors for cutting. But attach something called a deaminase. Now you can change a single letter of DNA without making a cut at all. Attach a polymerase, and you can write whole new sequences into the genome. Or attach a fluorescent protein, and you can just light up a gene to see where it is. It's one platform with really endless applications. So, this incredible power, it forces us to face some of the deepest ethical questions of our time. The ability to program the genome means that we, as a species, have to decide very, very carefully what programs we should actually be writing. Now, the single most important line we have to draw is this one, the difference between somatic and germline editing. Everything we've talked about so far, all those therapies, that's somatic editing. It affects the body cells of one patient. Those changes die when that person dies. Germline editing, on the other hand, that means editing sperm, eggs, or embryos. And those changes, they become heritable. They get passed down to every future generation, forever altering the human gene pool. For scientists, this is the brightest of bright red lines. And look, these risks are not just theoretical. The CRISPR baby scandal back in 2018 showed everyone exactly what happens when you move too fast. There's a risk of off-target edits, where the system makes a cut in the wrong place. And with embryos, you can get something called mosaicism, where the edit only works in some of the cells, creating a biological patchwork. Proving that a therapy is 100% safe with zero side effects, it's a monumental challenge. So, where does this leave us? We've gone from simple scissors to a genomic GPS to a full-blown programming platform. The destination isn't just about cutting out a few diseases. It's about having the ability to precisely and safely write and rewrite the code of life itself. The question isn't, can we do this anymore? The question is, how will we choose to? What programs will we decide are worth running?